Morning. I'd like to welcome all the PARC members and also guests and also people who are online. We have a number of people joining us online for our seminar today. I'm very pleased to introduce Dr. Greg Scholes from the University of Toronto as our PARC speaker today. Greg is a professor in the Department of Chemistry, and he's also the director of the Center for Quantum Information and Quantum Control, and a visiting professor at both the University of Freiburg and Beijing Institute of Technology. Greg is a native of Australia and got his undergraduate and PhD degrees at University of Melbourne, and then went to Imperial College London, where he worked with David Phillips, and then did another postdoc at University of California, Berkeley with Graham Fleming. So he has really outstanding credentials in laser spectroscopy. Greg's, I won't go through all his awards and so on. He's an editor, senior editor for the Journal of Physical Chemistry, and I'm very pleased to announce that he's recently joined the PARC Scientific Advisory Committee. And so we're very pleased to have him as part of the PARC group. Greg's interests are extremely broad. He really covers a whole wide range of issues in chemical dynamics, but in a whole range of systems, including nanocrystals and polymers, and also in biological systems, in particular photosynthetic light harvesting complexes. And so he really brings a broad perspective to that study of dynamics initiated by light. And so that's what he's going to tell us about today, lessons from nature about solar light harvesting. And the earlier version of it had a kind of a subtitle, which was "A Little Coherence, Please," or something like that, which I think most of you will get that joke, or if you don't now, you will by the end of the talk. So Greg, welcome to St. Louis. It's a pleasure to have you.
Thanks very much, Bob. Thanks for the invitation. Thanks for the introduction. Really looking forward to seeing or meeting with the people in the park here. I've already seen some pretty spectacular facilities. So let me get into the talk. I did tailor this talk specifically for this audience. I know you have background in photosynthesis, and I know that when Bob invited me, he was very interested to learn what all of this coherence stuff is about. So I have something at the end where I hope I can explain this to you somewhat clearly. In the big picture here, what always amazes me by photosynthesis is just the sheer scale of this solar energy conversion. 100 billion tons of biomass produced annually in water as well as on land by photosynthesis. To put that in numbers we can understand, that's every single hour of every single day, same mass of biomass as two great pyramids of Egypt. So this is a lot of solar energy conversion, as I'm sure you're all familiar with here. As Bob mentioned, I'll talk about light harvesting. And just to remind you how to think about light harvesting, this is a map, as many of you will have seen before, of the proteins in the thylakoid membrane of higher plants or green algae around about photosystem 2. This is the photosystem 2 complex. Both of these are the same. So the goal is to photoexcite that special pair, and that initiates charge separation. That's the solar cell in photosynthesis. But what's remarkable is the number of other molecules, other proteins around that reaction center that don't separate charge directly. All they're here for is to harvest light. It's completely different to how we design an organic photovoltaic, for instance. But this is what you can do when you have molecular level precision in your engineering, as you do in biology. So these other proteins here surrounding the reaction center of the light harvesting complexes. And what I want to talk to you today about is how the energy gets from where it's absorbed in one of these proteins to the reaction center. So light could be absorbed anywhere in these proteins, and there's going to be a series We might think of it as jumps of excitation energy. So not electrons, but electronic excited states of molecules are transferred through space. And this happens under low light conditions with very high efficiencies in all of these systems, greater than 90% efficiency. And that means for antenna systems of this size, the energy transfer steps fundamentally must be very, very fast, 100 femtoseconds or so on average. And it turns out that this makes it interesting from a physical chemistry point of view. That's what I want to talk to you about. Light harvesting complexes are, in addition, wonderful to study because they're so diverse. They come in all sorts of shapes and sizes. They all work the same, but the diversity is interesting. Here's some of the different structures I'm sure you're all familiar with. I've seen some of them already today as I've met with people and their corresponding absorption spectra, from the near infrared right down to the blue-green, depending on where these organisms live. And one of the things I've been thinking about recently that is a great illustration of light harvesting is sometimes it's not easy to understand why there's this diversity when you just study one of these proteins in isolation. You don't get the full picture. So the full picture only comes at the level of an ecological perspective in many cases. I really like this example I found in the literature of these particular microbial mats dug out from a marsh in Massachusetts. These are the mats here. It's about 1 centimeter thick that you see. And you see the different colored stripes through this mat here at the bottom of the estuary. These different colors are layers of organisms. And so they're translated actually over here in this diagram. Where you see at the top is oxygenic photosynthesis in the first couple of millimeters, diatoms and cyanobacteria, and then we go down to purple bacteria and green sulfur bacteria. Let me give you an illustration of what's going on here. Why is there the layering, and what does this mean? This is the solar spectrum incident on top of that microbial mat, and then it's measured after it passes through these layers with the oxygenic photosynthesis. That's that transmitted spectrum. You see what's missing here is everything in the visible and the red, chlorophyll, phycobilosomes, eating out all of this solar spectrum for their photosynthesis. So when this spectrum is incident on the next layer, that's the purple bacteria, which are absorbing, of course, at 800 and 850 nanometers. They're quite happy to photosynthesize here, and you see the spectrum transmitted through that layer, the pink layer, missing all of that light. So what happens next? The next layer is also purple bacteria, but now instead of bacteria chlorophyll A, they have bacteria chlorophyll B. It shifts their absorption further to the near infrared so they can photosynthesize out here. And then what's transferred finally down to the poor green sulfur bacteria, this trickle of photons here in the mid 700 nanometers that they photosynthesize on. And when you work out how many photons that is, that's 300 photons per second per organism in green sulfur, for the green sulfur bacteria. That's equivalent to a moonlit night on Earth that they're living under. And sometimes, especially in physics, this is remarked on as the amazing thing about photosynthesis, and we have solar energy conversion under moonlight, etc. But you need to remember the goal here is very different. Humans are greedy. We need to make lots of electricity, lots of power. These guys just need to survive. And they're reproducing, of course, but only every 3 to 26 years. And Bob and his group know infinitely more about all of these stories than me, but I think it's really nice to have this ecological perspective. But the bottom line here, as I move into more of how to think about energy transfer, is these light harvesting systems are really here to concentrate excitation energy. One of the things the graduate students can think about if they wish is this is especially important where you need multiple photons in sequence, not coherently but incoherently, to do the photochemistry like you do in photosynthesis. In this case, Poisson statistics come into play, and it's even more important to concentrate the excitation. I'm going to leave that as an exercise for you to work out why that's the case if you're interested. One of the things that you notice in terms of where light harvesting complexes are more important, it's well known, is under conditions where light is a limiting factor. Here's some micrographs of cryptophyte algae, which I'll tell you more about later. These ones are grown under high light, these under low light, these are intermediate light. Now I might just point out to start with, by high light actually mean, I think this is only something like 20 micro-Einstein per meter squared per second. So this is high light maybe for these cryptophytes, but yeah. And this is down at 1 micro-Einstein, which is thought to be the threshold for primary production. But notice that the different color in each of these cases. The color is simply telling you how many light harvesting complexes there are per cell in each of these cases, and quantified here in the absorption spectra of the whole cell. It's actually the ratio. What's changing is the ratio of the reaction centers absorbing here to the Microbal protein antennas. And so when you grow under low light, you have many more antenna per reaction center. It's much more important to concentrate the excitation. So there's a biological example. In chemistry, also very common to make supramolecular complexes of all sorts of varying degrees of complexity that can also concentrate excitation. These dendrimers, for example, light is absorbed in any of these chromophores and makes its way with 100% efficiency to that central chromophore. There's many, many examples here. And that excitation is funneled with a gradient of energies of these molecules according to Foerster theory, which is often what happens in photosynthesis as well. And I want to talk to you about that, the way that the energy jumps from molecule to molecule through space. But I first of all want to tell you why this is a hard problem. If we just think about Foerster theory, it's not such a hard problem. But let me show you this example. Here's an absorption spectrum of naphthalene, the S3 transition of naphthalene, very intense absorption spectrum. We could make this molecule, which is 2 naphthalene molecules. So now we would expect to be able to absorb light twice as easily with this molecule. And I can ask you the question, what will you expect the absorption spectrum to look like? And according to Foerster theory, actually it has to be the same as this. It would just have twice the extinction coefficient. If we measure it, it's utterly different. And this is a classic example of an exciton splitting. And here you are seeing quantum mechanics in the frequency domain. What these two peaks are that you have with this molecule here are the quantum mechanical superpositions of absorption on the left and the right side of this molecule. So one is the symmetric linear combination of excitation on the left and the right, and the other is the antisymmetric linear combination. These are Frenkel exciton states. So now the states of this molecule mean that absorbed light are states where light is shared quantum mechanically. It doesn't jump from one side to the other at all. So here's where the problem arises. At what point can we say excitation is uniquely sitting on one molecule so that it jumps through space? It does this in a limit of weak electronic coupling. We usually think about that as the case where this splitting here, which is proportional to the electronic coupling, the dipole-dipole coupling in Foerster theory, and that splitting is smaller than the natural line width that you see in that red curve, we would normally say Foerster theory works. But there's never been an experimental way to examine this, and that's been the problem for a long time. So this is what we want to do. We want to ask a question, not just in that static picture, but in a dynamical context. How does energy move through space? When is it shared between molecules? When does it jump from molecule to molecule? So we kind of need to make a map of what happens as a function of time after we put excitation in the system. And this is what this 2D spectroscopy does. And there's some special features in the map. Let me, first of all, explain how to look at this. I'm going to show you not too many, but a few pictures that look a bit like this, where we're going to have this excitation axis, in this case on the left-hand side, and the x-axis is the signal or the measurement that we make after some time delay. And what that does is it maps the absorption spectrum in 2 dimensions. So if we photoexcite, say, this part of the spectrum up here, this green line, and we observe it sometime later to observe the population of that state, we could follow the kinetics of loss of excitation in that state. On the other hand, if we wanted to look at this cross peak, we could actually watch excitation after we put it into this green line. We could watch it rise, fall down, if you like, into this lower energy green transition. So we could watch that cross peak rise with time. And so this is the kind of map I'm talking about. But there's going to be some interesting additional features that we'll see in this map, and this is what the coherence is about, which I'll mention to you in a moment. So here's some typical maps. Each one of these illustrates just one way to think about these two spectra, get you a little bit familiar with 2D spectroscopy. Here's the waiting time after I put excitation into the system. 0 femtoseconds, and I make a map after 30, 160, 1 picosecond. This is for a conjugated polymer. So you see, as usual, molecules in the condensed phase, especially complicated molecules, give broad featureless absorption spectra, and to some extent that's reflected in the 2D spectra. On the other hand, even this very, very complicated polymer, look at the shape of this line as a function of the waiting time after photoexcitation. We're growing in a cross peak. And what that is excitation transfer along the polymer chain downhill until it reaches the long red absorbing conformational subunits. So that's energy transfer you see in the cross peak. On the other hand, for these nanocrystals, this is a completely different looking 2D spectrum. And there's clearly two absorbing states. You see it on the diagonal there and there. They're the two lowest energy exciton states of the semiconductor nanocrystal. What's striking is the fact that you also have these really strong cross peaks. So this whole spectrum looks like a square. What this is telling you, in NMR, it's often called coupling. It tells you these two states are coupled. What it really means is that the photoexcitations of these two excitons involves common orbitals. In fact, what happens here, this lower state here, we excite electrons from one valence band to a conduction band. The upper state, we take electrons out of a lower valence band, but we put them in the same conduction band. So if I photoexcite this higher state, I put electrons in the conduction band, and then I probe the lower state. So looking at this cross peak here, I see that it's harder for me to put my electrons up there because you get something called Pauli blocking. So it tells you that the states are related spectroscopically. And that example is important because this is going to be the foundation really for how we probe this coherence. How we do it in the experiment is this. Sometimes easy to see by example. It's a femtosecond laser pulse split into two. And one of those pulses will be delayed on a delay stage. So this is going to be a classic pump probe configuration. One of the pulses is going to photoexcite the sample, and the other will probe. And so this is a lens, and in pump probe spectroscopy, we would now focus into the sample. A pump pulse followed by a probe. These are broadband, about 10 femtosecond pulses. But instead, this is something called a diffractive optic. That's going to make a perfect copy of these pulses and their electric fields. And so we're going to have 4 pulses, and we're going to need to do this to generate the 2D spectrum and to measure the amplitude of the signal. We want to characterize the electric field. We're not measuring an intensity on the detector here. We have these 4 beams. And what we need to do now is take 2 of them and put in this variable delay. That's the variable we're going to Fourier transform over later. We do that by pushing these wedges together. And then one of the beams travels through thicker and thicker piece of glass, and it delays it with sub-attosecond resolution, actually. The key thing is it doesn't upset the electric field of the laser pulse. And that's what we really need, these phase-locked pulses. So now we have a pulse sequence. This is a known field that we'll use to measure the signal. There's the 2 pumps and the probe. That's the sample. And we'll generate something called a photon echo, which is another little signal, looks like a laser beam, that will overlap there with that known field in the detector and spectrally dispersal of this. And on the CCD, we measure an interferogram. The frequency spread is one of our axes in the 2D spectra. We measure this as a function of the time delay between the first two pulses and Fourier transform with respect to that variable, and we'll get those 2D spectra I showed you. So this is a non-trivial experiment, but turns out, as I'll show you, to be well worth the effort, and is getting easier and easier to do these days. I'm going to apply it, I'm going to show you an application to these light harvesting complexes from algae. These are cryptophyte algae we cultured in Toronto. I showed you a similar species before. You see the red color, that's the light harvesting antenna, which we isolate to look at. I'm sure in PARC, again, you're aware of how important photosynthesis is in the oceans and in freshwater, and it mostly happens in microscopic algae and cyanobacteria. Algae are much more prolific than you'd imagine, even these cryptophytes. And if you take a liter of seawater or lake water, on average it will contain 1 million photosynthetic algal cells. Microscopic, like these. These are about 10 to 20 microns across. So this is another species. You see these are blue. They have a different color. Light harvesting complex, and that's the complex I'll show you these experiments on. This is a TM micrograph of a cross-section of this cell where you see the stripes here, which is a thylakoid membrane side on. And the light harvesting complexes are in the lumen between these membranes, and we can isolate them in buffer solution. This is this PC645 antenna protein from these cells here, or this is the red one that was isolated from the cells I showed you on the previous slide. We can study, there's actually more than these two colors as I'll show you before. And here's the plan. We know the structure of this protein, or this is a different one, but we know the structure of several of these proteins to sub-angstrom resolution. There's 8 light-absorbing molecules. It's about 5 nanometers across this protein. There's these 8 billion chromophores bound in the protein. Center to center, nearest neighbor distances are about 20 angstrom, quite far apart for a photosynthetic protein, actually. And the goal is, given that we know the structure, given that we can in principle calculate and predict energy transfer, we might think pretty well, we'd like to do an experiment that would map the energy transfer dynamics. And here's one way you could think about it. This is work we did with Renk van Grondelen. His group photoexcite the central part of the protein here on the blue edge, and we can work out how fast energy flows to the outside of the protein and where it flows. So this is a map of kinetics. And this is typically what people have done in femtosecond spectroscopy and biophysics for a long, long time. And this is important information. But is it enough to really understand the mechanism of the energy transfer? And actually it's not. This is not enough because this is assuming classical probability laws. We can't test a quantum mechanical model for dynamics with this information. It's certainly not a strong test anyway. What we need is not probabilities for moving through space. We need amplitudes. And I'll show you that at the end of the talk more explicitly why that's important. But it's amplitude information you need because of the wave nature of the quantum mechanical theory. So how do we get that? Turns out we can get information about it first. Probes of it, in any case, from 2D spectroscopy. And this is what's made the application of this experiment to light harvesting complexes really exciting since 2007 when Graham Fleming's group first showed you could see these so-called electronic coherences. So how do we see this amplitude level information? How do we see electronic coherences? I'll tell you right now, there's still a lot of work to be done in understanding what you can get out of the experiments and how you can take them further. I just want to tell you, make an analogy here and tell you what we're going to look at for the remainder of the talk. And that is things that oscillate as a function of the pump probe time delay in these maps. That turns out to be the interesting thing. So why do they oscillate in this case of electronic coherence? Come back to this picture I showed you before of the absorption spectrum. Each of these colored curves colored lines is a resonance. This is the center of the absorption band attributed to each of the 8 villains found in this protein. We know this from many other experiments. So you can think of these as these 8 electronic resonances. So if there were musical resonances, we'd know if we ring this note, we'll hear a high-pitched musical note. If we ring this green one, it's a lower-pitched musical note, and so on. And that's kind of what the experiment is doing. It's ringing these resonances, and we're listening to them, in this case, on the diagonal. But one important difference, this is a femtosecond laser pulse. Its bandwidth covers this whole map here, which is a large part of that absorption spectrum. It's ringing all of these notes at once. And if they were tuning forks, and we hit them and we listen, we hear the musical notes, but you'll also hear a low frequency beating at the difference frequency between these notes because of the wave nature of sound on the detector, in this case your ear. This experiment works in an analogous way, not actually exactly the same, but you can think of it like that. Because we photoexcite superpositions of these electronic resonances, as we wait after the pump sequence, we're going to accumulate a phase. Just comes from Schrödinger equation, but the phase gives a beat of amplitude in these spectra, and this is what we're interested in. So this is what's, I guess, important about the 2D spectroscopy from this point of view. This is the first time that we could really ask questions about quantum effects in the dynamical evolution of any actually chemical, light-induced chemical reaction. This is what you see. And this is a movie. It's a 2D spectrum from that blue protein as a function of waiting time. You especially see this feature up here beating in and out. This is going for hundreds of femtoseconds. We see a lot of other oscillations in this 2D spectrum as a function of time. This is room temperature sample of this algal light harvesting protein. So this was completely unexpected when we first saw this. So let me tell you a little bit more about what you saw in that movie that goes by really fast. The axes were different in the movie, by the way. So the cross peak that was up here, I've now changed it back to this normal axis system and put it down the bottom. But you can see there's been some huge improvements with the experiment recently. My postdoc, Duffy Turner, developed a new way of doing the experiment with several important modifications. What you're seeing here are raw data, just Fourier transformed on a linear intensity scale. And this is the beat, which I'll show you is electronic. Here it is at 25 femtoseconds with its maximum amplitude. You see it disappear, comes back at 55, goes away again, etc. That's the main beat in the cross peak. That's a superposition of transitions of the blue and the green resonances I showed you before. These are chromophores, which are in the middle of the protein and on the edge, so a couple of nanometers apart in space, which is perhaps more remarkable. And these kinds of observations have led to all sorts of news stories and speculations about quantum effects in biology. Some of you may have heard about this stuff, quantum life, etc., etc. Because we're seeing signatures of clearly a quantum mechanical effect in something taken from biology. But of course, the story is not complete, and what I want to describe to you now, and the rest of the talk is really what has to be done and what we are doing to ask the question: does any of this matter for biology? And if so, how should we understand why it matters? First thing is, we needed to work out if these truly are electronic beats unequivocally. There's a lot of people who are saying, well, can't this be a nuclear beat? Vibrations of the molecule, you know you excite these with short laser pulses and they beat. And I'll show you the answer is much more interesting than yes or no. I'll show you that now. But to answer that question, we had to, as I said, make this experiment work better. And this is what Duffy in particular played a big role in. Many people in my group have made contributions here. So what he's done is he's developed this 2D experiment That's what I like, is this precision of his wedge calibration to 100 zeptoseconds per millimeter of stage. This is incredible, and you needed to have this stability and reproducibility so that as we scan the laser pulses, we're not rolling the phase of their electric fields at all, because that would look like beats. You have to have this beautiful characterization. Here's an interferogram, what you see, interference, that's enabling us to reconstruct the signal, the complex valued signal. It's a function of that waiting time. You see these straight lines here for this laser guy, showing he's got this rock solid calibration. He's solved a problem here, a phasing problem. Similar problem you have in crystallography, an unknown phase issue. And that's what made the difference. So now, much to the pleasure of theoreticians who are always asking why our experiments always look different, here's error bars on these data. This is the amplitude of the signal I was showing you at that beat. This is you're seeing here a plot. It's a function of the waiting time of the amplitude of the beats. The different curves here are actually that same Billy protein, PC645, but a different pH. Because it sits in the lumen, we were wondering what happens under photosynthetic activity when the pH in that lumen can drop by 2 units. Could the protein regulate photosynthesis? It turns out it's completely robust to this pH change. You see these beats are identical. We can show that quantitatively. And from analysis of data like that, Duffy could work out a couple of important things. One is, first of all, you saw actually just by eye here, there's not one frequency here, there's not one phase that would correspond to the energy difference between 2 electronic states. There's multiple frequencies. And these are what they are. He found that there was 8. 2 of them are the dominant ones that we see by eye in the movie. And they're here in terahertz, which he likes to use. So this is 21 terahertz is about 650 wave numbers. 26 terahertz is about 800 wave numbers. These are the 2 dominant frequencies. And what I'll show you is that the 21 terahertz frequency is the electronic coherence. And at room temperature, its dephasing time. So assuming an exponential decay, 170 femtoseconds. So you're seeing this electronic coherence for approximately 3 times that time. This is quite striking. And the 26 terahertz which lives for 300 femtoseconds of dephasing time. That's vibrational. And the fact that they're together here, I think, is not a coincidence. I think this is important. I'll tell you more about that in a moment. But first thing, let's go through these Feynman diagrams. Only joking. What Duffy had to do to work out the difference here was tedious, but not impossible. He had to think about every way you could generate a signal from vibrational coherences versus electronic coherence. And he wrote them all out fastidiously here and color-coded them. And he could show that the difference being, with electronic coherence, you have 1 ground state, 2 excited states. In vibrational coherence, you have 2 ground states. This is the vibrationally excited ground state, and 2 levels in the excited state. And he could show that there were 3 ways of telling the difference between these models. One is, and I think I'll show it to you in the next slide, the main one that I'll show you is to look at these cross peaks in a decomposition of the 2D spectra, actually 2 half Fourier transforms called rephasing and non-rephasing spectra. And he could show, yes, good, I can show it to you right here, that in the rephasing spectra, you should see beats both for the vibrational and the electronic coherence cases. But in the non-rephasing spectra, you would only see it for the vibrational coherence case. Now, let me first of all show you the traces here. You see there's actually beats in both the rephasing and non-rephasing cross peak. When you Fourier transform them, however, we concentrate on those 2 frequencies I was talking about. So perfectly within the noise of this experiment, you see that there's this 21 terahertz frequency only in the rephasing data. And that's the test that it's electronic coherence. There was a second test, which gets more technical, I won't go into, that could verify that. And the frequency that you see in both spectra is vibrational, or at least has a vibrational component. That's as much as we can say. So now we have a clear test. There is for sure electronic coherence. So two more things I want to finish up by telling you. One is, first of all, this quantum biology business. We don't yet know if any of this matters for biology. Of course, we're studying isolated proteins. I want to say a little bit about that, how we might tell the difference, and then a little bit about how to think about what this coherence is and why it's a very subtle effect. One of the interesting things about cryptophytes is they come in all these different colors. They have many different antenna types. What I've plotted here is the number of cryptophyte species with a particular antenna type as a function of time millions of years ago. So the oldest, or the root of the tree for this clade here is a cryptophyte with PE545 antenna complex. And you see with time the number of species with that antenna complexes rises like this. That's that antenna here. And then you see millions of years later another complex was innovated, and that led to a series of species. That tells you that that these antenna complexes are an innovation. They provide some fitness to an organism or allow it to live in environments that it couldn't live in before. They're not just, it's not random whether I'm a blue or a pink or purple antenna. This PE566 was innovated much later. There's different chromophores and it gave rise to many, many species. So it gives a clue here that we can ask questions about the antenna diversity. If we could compare these antennae, we would learn something. If we could compare, if we could ask questions about combinations of chromophores that are not present in any of these organisms, we could explore unfit light harvesting complexes. So there's ways here systematically to ask about connections between speciation and evolution and potentially even electronic coherence. To do that, you need to do a lot of different things all at once. One of them is sequencing these light harvesting proteins. That's what we're doing with Beverly Green. And there's some aspects to this that I'll discuss with people afterwards that I think are rather interesting. There's at least 3 genes for each of these antenna complexes. It's not obvious why that is. And these are the complexes that we've cultivated in Toronto, and actually almost all of them now we've studied with 2D spectroscopy. Of course, I don't have time to tell you all about this, but I think it's by comparing many, many species in this way that we can start to learn about the differences and similarities and try to translate that back to the biology. This is a big project. But what about the coherence? And is coherence really important? Do we need it for light harvesting? That's a contentious question. I think it's the biggest driver for these structures. Probably the most amazing optimization is the concentration of chromophores in these complexes. You take the classic example of LHC2, 0.25 molar concentration of chlorophyll. If you made a solution of chlorophyll 0.1 molar even, it's completely non-fluorescent. Excited state lifetime is picoseconds. You have no light harvesting whatsoever. So it's the arrangement of these chlorophylls, the perfect center-to-center separations and orientations in that protein scaffold that are perhaps the biggest innovation in these systems at all whatsoever. And it may be simply driving the system to have high concentration of chromophores that inevitably leads to a case where first a theory breaks down and you're left with having some coherence. And it may not be the coherence itself that's being sought, but we don't know the answer to any of these questions. Let me explain just one step further. This plot here, I have electronic coupling, which you can think of as distance between the molecules. and energy transfer time constant, just with a simple, the ideas that Förster spoke about many years ago. So you'd expect that as you make the electronic coupling bigger, the rate of energy transfer increases, something like this. You get this turnover to 1 cubed distance dependence. And so you could just pack the chromophores more tightly, subject to concentration quenching, and have faster energy transfer. But something happens over here, and that's what I showed you earlier. Strong electronic coupling, you get these new spectroscopic states, excitons. And so now you don't actually have energy jumping from molecule to molecule, but it's a radiationless transition just to bridge that gap. So in fact, you get this curve here where we correct for that, and you get a turnover to exciton relaxation, which gets slower with bigger electronic coupling because of the energy gap law. So there's some peak here, which is the sweet spot, I suppose, for optimal energy transfer times, and therefore longer diffusion lengths. Important for low light photosynthesis. And we don't know much about that peak, but clearly it's in a regime that's halfway between excitons and Förster, so this is where you must have coherence. And in the last part of the talk, so this is what I have especially for you here at PARC, is to give you, I'm going to show you some equations, I think on the next slide, which I'm then going to turn into a picture simply based on this Förster spectral overlap. I'll explain to you why coherence is really, really improbable from the point of view of Förster spectral overlap. So you're all familiar with the overlap, that it's the donor emission spectrum overlapping with the acceptor absorption that gives you this spectral overlap, and the rate of energy transfer is proportional to something to do with electronic coupling times that spectral overlap. And the Förster limit is a very powerful tool, and this is why Förster theory is so popular. These two pictures I'm going to flash through. What my former student Kathy Wong did is worked out how to subtract the slowly varying background from the 2D spectra that I showed you already, so that she could obtain just a picture shown here of the part of the spectrum that's doing the beating, the electronic coherence. So she could ask questions about the shape of those features, because this is something else I didn't mention in 2D spectroscopy. The shape of these features tells you a lot. And I don't have time to tell you about it, but you can watch these features here. You see the bottom, let's just look at the bottom cross peak going blue, red, etc. across here. That's the oscillations from negative to positive that you have with this electronic phase. In any case, what that told us is that there's correlations in the system that are actually rather unusual, and that mean that you can't make that nice separation of electronic and nuclear factors that Förster did. So the spectral overlap becomes hard to deal with. And that's tied into this theory that I'll show you, but we don't need to talk about it any further. Whole point here, coming back to my amplitudes, how do we think about electronic coherence in the simplest way? The simplest way would be to start from Förster theory and add a little bit of coherence. And in that way, we can keep this picture of amplitudes, and I can show you why a quantum mechanical theory is different than a classical theory. So you get these, from solving Schrödinger equation, you get these time-dependent coefficients, which we can expand in a series called the Dyson expansion. And they're going to carry information about the paths that energy transfer would take at the amplitude level. The transition probability, then, from going from the initial to final state is the sum of these different paths, and then we take the modulus squared of the sum. So these are like, if you think of them as waves, you're summing the waves before you take the intensity. Compare this with the classical law, where we would just take the paths themselves as probabilities. We won't have the interferences that we'll have here. For instance, here we're going to have one path, we're going to have a term that's one path multiplied by a second completely different path. That's an interference of the waves, so this term times that term. That's what makes the quantum mechanical description different. And let me show you why the probability of these pathways just diminishes rapidly as you have more and more complexity in the possible paths. That's the point I want to get here. You can develop a theory for this. As I promised, I'm going to show you a picture on the next slide that tells you why this is, but you need to get an expression for the rate here, and then we can have these factors that tell us about those coefficients in the rate. We have a summation here. So this is Förster theory. This one here, so it goes down here. That's Förster theory. This G2, this is a second pathway. This is where we go from donor to an intermediate molecule to a final molecule. That's that pathway here. This was too complicated to work out the full expression for it. This is the interference term of those two pathways happening simultaneously. And that's what our coder focused on. And the difference between all of these essentially is the spectral overlap. And what's particularly important here, there's two factors that allow you to make this expansion and not have these higher terms, the coherences go to zero. Two things you need. And actually, I'm going to skip the static picture. Well, actually, let me explain it here. In Förster theory, we could think of these free energy curves like this. Chemistry, we're always interested in drawing potential energy or free energy curves to think about activated chemical reactions. So you need to get to that transition point, and then you can cross the barrier. This is what Förster theory is, but it defines that curve crossing there as the point where the donor emission spectrum is equal energy to the acceptor absorption, because that's exactly what that is in this reaction coordinate for Förster theory. A little bit more complicated, but this is the cartoon version of it. So that's what the spectral overlap tells you. When you have coherence, the same thing. That crossing point is the important part. That's when the transition energies of the molecules are equal. That's when you get excitons. I showed you that with the naphthalene picture. That's when the electronic coherence is most important. So what we need to do to get energy transfer in the Förster picture, the fluctuating transition energies of the molecules like this, they become equal, then there's some chance of energy transfer. So we have to wait for that to happen. So we need this energy resonance, and we need some temporal correlation. We need that point where the energies become the same, not to happen in 1 attosecond and then disappear. It needs to be around that energy for long enough to get energy transfer. So we need these two conditions. So here's how to think about what governs how many molecules can participate in coherence. It's like the spectral overlap, but now we have to have, say, the interference term, 3 molecules in overlap. And you can think of the area here as the effective spectral overlap, where you've got to get 3 molecules at the same time to have their energy gaps equal before the fluctuations take them out of resonance. If we have 4 molecules, we need to have 4 molecules fluctuating and get them all in resonance for the time of that whole energy transfer. It's like juggling. You've got to get the 4 balls in the air at once. And so this becomes exponentially harder and harder to do the more molecules you need to wait to get together, especially if they're all fluctuating independently. So this is in the weak coupling limit. This is how to think about why we don't expect to see significant coherence. And when we do, the correction to Förster theory is small. Of course, it's different when electronic coupling is strong, as I showed you in the excitons, and then that energy scale dominates the fluctuations of the energy gap amplitudes. So I didn't really answer the question as to the importance of coherence. I think it's a pretty subtle effect. I don't think it's something that completely transforms the efficiency. I'll show you this one slide to finish with, just to tell you that 2D spectroscopy isn't all about seeing coherences. It's also about thinking about spectroscopy and energy transfer and pathways of energy transfer. And here's an example from LH2 from Rhoda-Pseudomonas acidophila in collaboration with Richard Conkle's group. This is the absorption spectrum of the complex, of think you're all familiar with it. The bacterial chlorophyll absorption is the QX, and then the intense S2 absorption of the carotenoid. Did a number of experiments here. I'm just showing you one 2D spectrum, where what my postdoc Evgeny did is he tuned the laser pulse so it covered just this part of the spectrum here. It avoided the S2 transition because that bleach dominates everything if you cover it with the laser pulse. And he did that, and he sees on the diagonal this peak, and then here's the QX of the bacterial chlorophyll. So he's seeing that absorption there, and then something, we don't see an absorption here, right there. And through various analyses, he can show, for example, the beats at this point here. They're the carotenoid vibrations. This is a carotenoid absorption. And it's sitting right in here. And it has the hallmark, actually, of this 1Bu minus state. Optically, not quite forbidden, but close to forbidden transition that may be just here. More importantly, he sees a cross peak that connects that dark state to the QX of the bacterial chlorophyll. So it's telling you that this is an important energy transfer pathway, which suggests relaxation from S2 to this 1Bu minus mediates the energy transfer into the bacterial chlorophyll part of the ring. And this is something you can pick out, you see quite clearly in 2D spectroscopy, but in Pumpprobe, everything would be on top of everything else. It's much harder to pull it out. The information is there, but it's harder to pull it out because you also have this strong S1 excited state absorption negative feature that will cancel out a lot of this and dominate over a lot of these other effects that we see when we spread it in 2D. So that was just a last thought to show you what else you can use 2D spectroscopy for. So I've left you with more questions than answers, maybe. What is the role of coherence in biology, if any? No answer. Can we exploit coherence to design something? I didn't address that, but I think this is an interesting question. I believe you can. We've looked at this theoretically. And as I said, on average, and this is known from physics, in a noisy, disordered quantum system, these are physics words, it's a very well-studied problem. It's known that on average, coherence doesn't matter. But on the tails of the distributions, it can make a profound difference. And that, I think, is an interesting point to think about further. And I wanted to just, again, undo one of the things I said and leave you with a further question. I mentioned the driving force in these design of these light harvesting complexes may be chromophore concentration. I showed you LHC2, 0.25 molar. You could compare with PS1, at least 0.6 molar, or the cryptophytes, 0.1 molar. Quite different concentrations here. So this whole area is still wide open. We basically understand energy flow in these systems, but when you get to the mechanistic details and the why and how can we exploit it, there's more questions than answers. So I'd like to thank as many collaborators that have been involved in various parts of this work, but in particular my students and postdocs. I especially want to thank you again for the invitation to come here. With those symmetric, the naphthalene systems, we can actually, just in the frequency domain, spectroscopically, we can characterize what those transitions are from their polarization and from the strength of the two transitions. So you can work out, for instance, that they're two-level systems that are coupled, and because they're degenerate, it's easy. The splitting is twice the electronic coupling between them. So that's a more straightforward system. When you have much messier, more unknown things like the billy-proteins or an arbitrary system, this is much harder. And so you've got more energy scales at play. You've got the energy differences between the different molecules. So they're different two-level systems with different energy gaps. And then you have different electronic couplings among them. And then you have the coupling to the environment. It's another energy scale, and it's the interplay of all of these things. And all the energies are pretty similar here, typically in these photosynthetic systems. So this is the challenge. So even in the spin boson model, this is a really hard problem to solve. Oh, that's right, yeah. Okay. So it's a long question. So, yeah, I mean, what the question comes down to is how we think about electronic coherence. When we go from the excitonic system, if you think in the site basis, we automatically have delocalized states. So you would say in the site basis, does that mean that no matter how I excite the system, it's coherent? And this is basically Julie's question. The answer is it's more complicated than that, of course, in that the answer has to be independent of the basis that we use, especially when you think about incoherent light being absorbed. It's very different than the laser pulse. As you all gathered, I think, from the talk, the short laser pulse excites superpositions of these states. The laser pulse is making the coherence, which we can then follow. It didn't come from nowhere. So what we're doing there is we're not trying to see how the dynamics happen. We're probing the properties of the Hamiltonian that would decide what happens after incoherent light is absorbed. However, no matter what, when an incoherent photon, which is completely quantum mechanical, is absorbed by this system, it's absorbed into an eigenstate, and there's no coherence. However, it turns out that if you have memory exchange between the system and the bath, as dynamics happen, you can introduce these coherences with time. We have some, I don't have them with me, we have some simulations of this. And what happens is you need to exchange information with the environment, with the bath, and then it comes back to the system, and it can come back, doesn't have to come back to where it came from initially. And this can lead to the formation of superpositions, which can then be propagate with the dynamics. That's right. So a coherence, we would define coherence for those excitonic systems as a superposition of the exciton states, not as just one exciton state, which incoherent light could excite. This becomes horribly confusing and difficult to deal with, but in physics there's a much clearer understanding of what all of this, of how to deal with this. Yeah, so the question is about the role of, then when we add vibrations to this picture, how do we have to think about it? I think the key features of the system I showed you is that the energy levels of the bilins that are involved here, I showed you in the lines I showed you, blue, green, red lines, these are chemically different bilins. So they have different energy gaps. And if we go back in the presentation to first the theory, here, you can see when they, unfortunately this is the two identical chromophores, but if you imagine them being different, the purely electronic transitions are not in resonance. You won't have excitons in that case. So what would happen though, if this is the higher energy chromophore, if it de-excites to a vibration in the ground state, that is a perfect resonance then with excitation, purely electronic excitation of the acceptor. Or absorption of the acceptor to a higher vibronic state is perfectly in resonance with the purely electronic transition on the donor. So vibrations can introduce these energy resonances which help coherently share the excitation. So you then extended your question to timescales and so on. I think there's completely an interdependence of the timescales. And when I showed you the dephasing times of the electronic versus vibrational beats, they weren't too different. And I think that's not a coincidence. Of course, we have vibrational wave packets, but I think it's showing you as long as you can keep coherence of these vibrations, then you have these energy resonances that bridge the electronic gap. So it's like, I think it's the coherent version of Foerster's spectral overlap, essentially, that's going on here. So yeah, I agree with everything that you said.
So the different types of antenna complexes that these coherences have been observed in now, of course, first the FMO protein and now the bilins. Now, is that the limit, or are there others that have been demonstrated to show this? And I guess the question really is sort of heading toward, is this a general phenomenon? Are we going to find that there are some complexes that simply don't have any coherence type effects, maybe chlorosomes where you've got very strong coupling of pigments and so on?
Yeah, that's a good question. I think the bilins is a surprising one because it was, as I said, the most dilute concentration of chromophores. So that suggests that in various other complexes we would expect coherence. The Fleming group have a paper online now that's in the next issue of Nature Chemistry on LHC2 at 77K. It would be very interesting to do those experiments at room temperature. LHC2 has some pockets of strongly coupled chlorophyll, so does photosystem 1, so would not be surprised at all. I think it's almost certain that these complexes have coherence involved there. And you're right about the chlorosome. That comes back to this question of basis sets and so on. The electronic coupling is so strong, so I think that's one case where you have a clear, if you think of it as from the site basis, a clear quantum speed up of energy harvesting. You must have huge diffusion lengths of the exciton in that complex. But there are unpublished 2D results in the chlorosome where they don't see the beats. But again, you have one bright state and the other exciton states are dark, so you can't make the superposition and probe it anyway. But yeah, my feeling is once you get into the strong coupling regime, relaxation times between the exciton states, we know they're very fast. I think that dominates and wipes out the superpositions of the excitons.
In the chlorosome, the big issue is, I mean, pigment concentration is even higher than you were saying. It's over 1 molar, I think, if you just sort of work out the sort of average concentration of pigment, and how you can avoid the concentration quenching, because you've really got these things in intimate contact, and whenever you make the artificial systems, they all seem to be very, very quenched, even using the same pigment. So there's some sort of magic there that manages to avoid that quenching, but we don't understand that.
Yeah, definite magic in the fluorescent.
Any other questions for Greg? Okay, let's thank him again for a really
Thank you.
fun talk.